If there's one thing that puts me in mind of that lame first chapter of my ninth grade life science textbook, it's the eternal question of, what is life? This always gives me the impression of some kind of introductory throat clearing before getting to the meat and potatoes of what biology is about. On the other hand, over time I've realized that these basic questions about living things are actually not only interesting, but critical when we're looking at life at the margins, particularly during the early history of life on Earth. The same questions are also of interest when we are considering how much of a redwood tree is alive, or if a bacteriophage is alive, or if bacteria are alive when they're frozen, or even how we distinguish between people being dead or alive for the purposes of medicine or law. We all agree that living things have infested the Earth for billions of years, but identifying the actual start of life is where these questions become interesting, because there were billions of years after biological systems came into being before the first living thing arose. The origin of life on Earth, or how the universe began, isn't explained by Darwinian evolution, but that's okay. Evolutionary theory isn't intended to be an explanation for all things everywhere, just how life evolved once all the components of living things came together during the early history of the Earth. As cosmic dust accreted together and formed the solar disk, particles were drawn together by gravity, eventually forming a star at the center with 8 big planets and a host of asteroids, comets, and moons scattered throughout. Our own moon was formed about 4.5 billion years ago, about 30 to 50 million years after the origin of the solar system, when a body about the size of Mars collided with Earth and coughed crust and mantle into orbit, much of it coalescing into the stable moon over several million years. The early history of the Earth was dominated by the processes of volcanism that eventually formed the first continents. Water from comets and atmospheric rainfall accumulated into the oceans as the surface cooled. Working from first principles, there must have been a number of steps involved in the formation of the first living things. First, small molecules like atmospheric gases would have had been formed into larger organic molecules. Once organic monomers formed, then polymers of them could also start to accumulate in the oceans. Eventually, those polymers would have to be separated from the rest of the oceanic solvents to form early chemical reactions that could be contained within a cell. Though this sounds like it makes sense in principle, what's the evidence that such a sequence could have happened? Evidence of the plausibility of the prebiotic soup idea came through a series of experiments starting in the 1950s. Stanley Miller and Harold Urey developed an apparatus that heated and cooled different atmospheric gases of small organic molecules, sparking them to elicit the production of numerous larger organic molecules. All of this done abiotically. These experiments became more sophisticated as geologists' understanding of the early atmosphere came to light through the 20th century. But what these early experiments demonstrated was that the possibility existed that there could have been a period of time of accumulation of biological molecules on Earth. It's also possible, even likely, that Earth could have been seeded with biological molecules or organic molecules from space on comets and meteors. It is true that this kind of bootstraps the development of life on Earth, but it's also possible for novel kinds of chemistry to have taken place within comets or from meteorites cast off from other planets. Note that the initial atmosphere of Earth was probably a reducing one, meaning that there was little to no free oxygen present in it. Because oxygen wants to bond with everything, it tends to prevent the elongation of biological molecules, so the reducing atmosphere would have allowed a period of abiotic synthesis of organic molecules to persist for millions of years, until cyanobacteria polluted the initial atmosphere with free oxygen, which was a waste product of their photosynthetic process. It is also likely that the early production of organic molecules could have been assisted by the presence of clay minerals. First of all, when you think of clays, don't think of modeling clay. Geologists call clays these minuscule particles of silicate minerals that accumulate in sheet-like forms. When we look at the microscopic structure of clay, we see these numerous thin sheets that can be attractive to charged ends of organic molecules. So these clays could have acted like templates by stabilizing nucleotides on a surface, making it more likely to have productive chemical reactions that would allow them to polymerize. Today, living things use enzymes in order to catalyze such reactions, but in the early Earth, this wasn't necessarily an issue. Organic molecules could come together in the absence of enzymes if the temperature of their solution were higher than what might be tolerated by an actual living thing. It's also necessary to keep this early chemistry from being diluted before living things could really start. The evolution of protocell membranes from abiotically produced lipids could have started due to the self-organization properties of these amphiphilic phospholipids. In small concentrations, they form tiny spheres called micelles, but as they aggregate more and develop a water-filled space inside, they form up the characteristic bilayer that keeps the internal liquid and the chemistry of any organic molecules inside separated from the water outside. These early vesicles were probably made up of smaller and simpler single-tailed phospholipids rather than the double-tailed ones that are common today. These single-tailed lipids have been shown experimentally to flip from the inner to the outer surface with relative ease, so this might have been a way to bring molecules into the internal space of the protocell before the advent of cell surface proteins or pumps. How would selection for different kinds of protocells begin? As always, we need variation and differential success. As phospholipids accumulate, they tend to form tubular extensions off the initial vesicle. With some agitation, those tube-like extensions will split and form new bilayered vesicles. Growth is also determined by the internal components of the vesicle. Today, cells are complicated machines with a number of protein pathways needed for the care and feeding of DNA and using DNA to be the information to make proteins. It's more likely that RNA, accumulated abiotically, would have been the initial nucleic acid found in these cells. One important attribute of RNA is that it is autocatalytic and that one segment can serve as the template for the other side. In this example, RNAs can form by simple base pairing rules if the precursor molecules are present and a template is available. Where does selection come in? Cells that were able to accumulate RNA more quickly and that were able to amass more phospholipid would tend to be selected for compared to cells that did not have those characteristics. So, cells that were faster growing would tend to be selected for. These early genomes inside protocells were probably random ones, selected only for their ability to accumulate more RNA and to promote cell growth and division. How could more complicated chemical pathways have evolved? The idea of the hypercycle has been promoted to account for this trend. Briefly, the idea is that when chemical reactions can happen in an enclosed space like a protocell, the products of one reaction become the reactants of the next. This mental model is a way for us to imagine how the various chemical processes that are typical of cells today could have initially evolved. Chemical reactions inside cells that tended to promote cell growth or prevent cell dissolution or other attributes of cell survival would tend to be selected for. Once that initial advantage is had, it also needs to be weighed against the energetic cost for having that pathway present in the cell. Early protocells were probably not very good at their job, but those wobbly early steps towards cellular organization were tolerated because there was little other competition and because of a long period of accumulation of organic molecules for them to munch on. The early Earth was an RNA world, and it wasn't until much, much later that DNA supplanted it as the primary information-holding molecule in cells. One advantage that DNA has over RNA is its self-correcting nature, particularly allowing larger genome sizes. As the level of selection grew more intense for those early cells, then DNA became an advantage and became universal among whole cells. Today, when we reconstruct the early history of life on Earth, we suppose that there was probably some first living thing, some first cell that had all the attributes that we would apply toward living things. It's possible, even likely, that the first living thing is not a relative of anything alive today on Earth, though. Given the universal genetic code, we have strong evidence that all living things on Earth share a single, last universal common ancestor, a cell that either outcompeted or just outlived cells with other genetic systems. Taken together, the history of life on Earth probably started slowly after a period of millions of years of abiogenic accumulation of organic molecules. This soup of organic molecules became integrated into protocells at random. Early selection of these initial self-perpetuating chemical systems was probably based on little more than the ability of these cells to grow and divide faster. Early cells were probably also not very particular at keeping genetic material to themselves, so it's possible that there was accumulation of larger or more complicated genetic materials just from the soup alone. Where we head after these initial stages is to a period of increasing organismic complexity. In answer to the question of why life seemed to have gotten more complicated over its history, Stephen Jay Gould encouraged us to consider organism complexity as the measurement to consider. We know from our study of natural selection that living things would tend to avoid competition if at all possible. If the oceans of Earth were filled with protocells possessing random genetic codes, more complicated chemical systems would have been selected for on balance because there's more success to be had at a higher level of organismic complexity. Before the Cambrian period, unicellular life only achieved a certain level of complexity, about the level of a bacterial cell. It wasn't until after the evolution of the eukaryotic cell and then later the diversification of the major phyla of life in the late Proterozoic or early Cambrian that the next big jumps happened. So, most of the big transitions in the history of life have been caused by living things developing new phenotypes that allowed them to work together or work differently to better utilize an unoccupied ecological niche. You can pause on this slide if you want to read some of the details here. For the rest of this video, I'm going to focus on one of the major transitions of life that was symbiotic in nature, the evolution of the eukaryotic cell. Lynn Margulis was an American evolutionary biologist who was one of the major proponents of the theory for how eukaryotic cells evolved. The initial idea came from Alexander Oparin, a Russian biochemist, but Lynn did a lot of the primary work to provide evidence for the process. I met Lynn when I was in graduate school, and her approach to science and to life left a big impression on me. She was a spokesperson for the small, for looking at species that many of us might consider too simple, and to be able to tell us a story of what we can about the early history of life on Earth. She wrote, "Life on Earth is such a good story, you cannot afford to miss the beginning. Beneath our superficial differences, we are all of us walking communities of bacteria. The world shimmers, a pointless landscape made of tiny living beings." Today, the idea of the bacterial microbiome is a common one among evolutionary biologists, and we understand more and more about how the bacteria that live in us and on us contribute to our health. This is one part of the pointillism that Lynn talked about. Those bacteria help us to digest our food and in some cases can affect the way that we use that food energy. At what point is the boundary between their body and ours? The evolution of the eukaryotic cell probably happened in a few different phases. Today we envision it like this. First, a bacterium would have ingested another bacterium but not destroyed it through digestion. These two cells, now living together as one unit, had attributes that were different than they had when they were separate. We think that a bacterium, like a living thermoplasma, is likely to have been the host cell and housed the nucleus, while a bacterium similar to a spirochete would have given it the ability to have motility. In a second step, that host cell could have ingested an anaerobic Bellidivrios-like bacterium that could survive in aerobic conditions. The early Earth probably had an anaerobic atmosphere, as we said earlier, and the ability to function in the presence of free oxygen would have given an advantage after cyanobacteria had polluted that early atmosphere with their waste oxygen gas. This gave cells with those protomitochondria an advantage over bacteria that had to live in anoxic conditions. This basic combination of host plus spirochete plus mitochondrion is the basic cellular arrangement of both animals and fungi. Plants would have evolved with the inclusion of a cyanobacterium that would have given these early plants the ability to make energy from sunlight in the presence of oxygen. It is likely that they all have also lost their spirochete commensal. What's the evidence for all this? Recall that mitochondria and chloroplasts and even flagella have their own reduced DNA that we also use for systematic as well as forensic information. If these were parts of cells that just evolved within a single cell, it's not as likely that they would have had their own DNA. In short, these different combinations of eukaryotic cell types are found today because they allow advantages to many kinds of living things. Also, remember that all multicellular things are eukaryotes. The presence of these organelles inside the cells or their derivatives of them conferred benefits to their many ways of life. The pointillistic picture of the bacterial world that lives their lives in and on us extends past the microbiome to a consideration of the whole Earth. James Lovelock developed an hypothesis that he called Gaia, one that proposes that Earth is an actual living system. The life on Earth's surface is not just some infestation, but rather an essential part to the whole organism. He wrote, "You may find it hard to swallow the notion that anything as large and apparently inanimate as Earth is alive. Surely, you may say, that the Earth is almost wholly rock and nearly all incandescent with heat. The difficulty can be lessened if you let the image of a giant redwood tree enter your mind. The tree is undoubtedly alive, yet 99% of it is dead." That's a picture of Jim with Lynn. To pull things full circle, why the list of attributes of living things is interesting is when we are thinking about these kinds of questions. When do we declare that life really began on Earth, now that we understand that there had to be a long period of accumulation of biological molecules spontaneously? If only the outer cambium of a sequoia is living cells, but we think that it's alive, then could we accept that the Earth is alive too?